Okay, welcome everybody to another one of our KBase webinars here. Really appreciate to have as many people as we do on the line right now. My name is Benjamin Allen. I am an outreach coordinator for the KBase project. I'm based out of Oak Ridge National Lab in Oak Ridge, Tennessee. KBase is a multi-laboratory project sponsored by the U.S. Department of Energy, Office of Science. We have staff at Berkeley Lab, Argonne, Oak Ridge, Cold Spring Harbor, Brookhaven, and beyond that help us build this software platform. I think I said this earlier, but in case you're just logging in, we have a lot of people on the call, so we're asking you to write your questions into this document, the question and answer document that I shared with everybody over email. I'll send it around on the chat again. Just so you know, we have a lot of people on, about 125 or so. So just in case we have any bandwidth issues or anything like that, just stick around. If the meeting gets bumped or whatever, we're going to just use the same link to sign back in. It booted me for some reason earlier. Yeah, I'm sure like many of you, I'm also working at home, so bandwidth and those sort of things are very scarce resource, unfortunately. Yeah, you can write any questions you have down here, and folks will answer it. We are going to share the recording of this webinar afterwards. So if you signed up, we will send you the link to the recording pretty soon afterwards. So just, don't worry, everything that we tell you today, we're trying to make sure you have access to it afterwards. So yeah, look forward to that coming after the webinar is over. If you need anything immediately, you can write it into the chat, or if for whatever reason my voice goes away or something like that, you can write it into the chat. But for any major questions, we just encourage you to use this document. Okay, so let's go ahead and get started. I'm just going to start with some quick introduction about what is KBase at a very general level. We'll go over some of the basics of using KBase. So just, yeah, the very basics of the working environment. How do you create an account? What does the website look like? Where do I log in? And that kind of stuff. And then we're going to spend the majority of this hour and a half going through an example of using KBase to do some genomics research. So with that, I'll start up this presentation. So this is my email address here. Of course, if you want to contact me afterwards, feel free to reach out to me or to our main KBase account at engage@kbase.us. So introducing KBase. What is KBase? KBase we describe as a knowledge creation and discovery environment for both biologists and bioinformaticians. What that means is that we take data from all over the public reference data landscape, from JGI, from the data that's already out there about genomics or multi-omics data, and bring it into this system. We integrate it into our platform alongside third-party tools from developers that are in the open science community, and we bring them into a platform where those tools can be used to analyze both user data, but user data in the context of public data. So that's part of the power, is that we allow you to take your own data and understand it in the context of the public data that's out there. And on top of that, we build tools that help streamline that. So this is what we call our knowledge engine. So we're developing tools that enable you to make inferences or discover new findings in existing data. It's a very easy system to get your data into. We'll talk about this a little later, but KBase is really a drag and drop point and click interface. So it really lowers the barriers to bringing your data into the system because it's a very easy to use graphical user interface. So you can drag and drop your data and import it into your narratives relatively quickly for analysis. As I said, we have a lot of public and reference data in the system. So we have tools that allow you to search and identify that data, bring it into your narratives as well. And we also use tools like Globus to help manage large data sets. So perhaps you have, hundreds of gigabytes or terabytes worth of genomics data, and you're trying to bring it into the system. You can do this with Globus, which allows you to create essentially an endpoint on your computer or on your hard drive, and connect it to KBase and move your files over very seamlessly. We have all of these open science and third-party tools that we've brought into the system that do all sorts of things. These are some of the categories that we organize these tools into. So reads processing for next generation sequencing reads, assembly, annotation, sequence alignment, searching, comparative genomics, metabolic modeling, RNA-seq and expression data, metagenomics and microbial communities. We really have a lot of different tools in the system that allow you to take the data you have and analyze it through a lot of different pathways. And this map shows you what some of those pathways are or workflows, but essentially there are some common pathways that scientists often use KBase for, and this is what it tends to look like when you map it all out. This comes from our project paper, which was published in Nature 2 years ago, and that we'll talk about a little bit more later on. Everything you do in KBase takes place inside of what are called narratives. Narratives are Jupyter notebooks that we have retrofitted for use in KBase that allow us to contain all of the data, analysis, commentary, code, anything that is done on your data to analyze it. It's contained in narratives. And so these are reproducible documents in the sense that other scientists can get access to them. They can see where your data came from. They can see what parameters you use to analyze your data. They can have access to essentially your digital lab notebook that contains all of the work that you've done and all of the data. By default, all of your narratives are kept private, so they're just for you to look at. And you can, of course, share them. You can link your narratives in publications. You can share narratives amongst your colleagues if you're working on a common project, so that everybody can be operating in that common environment. This is sort of similar to how Google Docs and these other sort of cloud-based systems work. It's a little different, but essentially offers the same functionality in that you can collaborate in the same environment. And like I said, we are encouraging folks to link to your KBase work in your publication. So we've got over 100 publications now that people have used KBase to do some kind of meaningful science with. And we're increasingly seeing people do this, which makes us very happy. So we're encouraging all of you all, as you use the system, to link out to your narratives in your papers. Like I said, you can share your narratives with your colleagues, give them different levels of permissions. You can make narratives public. You can link to them in your publications. I'll talk about a feature later on called static narratives, which is a new feature that essentially allows anybody to access your work in KBase even if they don't have a KBase account. So right now, to look at a public narrative, you still have to have a KBase account, and you have to log in and so forth and so on. We now have the ability to create static narratives that you can link to anywhere, and they create a sort of saved state of the exact state of your narrative whenever you make it. And anyone can view it. So I'll demonstrate that later on. We also have organizations where you'll write, if your lab, if your institution has multiple narratives that they want to share, you can use organizations to bring those narratives and all of those users together into a common environment to do work. So I'll show that in greater detail later on as well. And yeah, so KBase is powered by two computing systems. Our main production system is based out of Argonne, and our sort of experimental and developmental platforms are based out of Berkeley. So you can see some statistics about the hardware there. From a user perspective, what you need to know is that KBase provides the compute for you. Anything you do on the system is not using your computer processor or RAM, except for running your web browser. All of the computation for your jobs is done on these systems. So it's a fair share type system in the sense that, if one user is running 100 jobs and another is trying to run 10,000, we try to allocate those in a fair way. But that's just to kind of give you some sense that we have a lot of people using these resources. We have to always figure out ways to share and queue and try to support everybody that's trying to use our system. And so sometimes, your jobs may run up to our maximum limit against the queue. So if your job hasn't started and executed after 7 days, it'll likely time out and you'll have to rerun it. But overall, we're seeing a lot of people using this system, and there are queues, but they are still shorter in many ways than a lot of what else is out there as a public offering. So, and this is some old stuff, but overall the use of the system is greatly growing. We're seeing way more publications, more data, more users. Okay, and on that, I'm going to stop the slides and go into some more stuff. So one thing I wanted to point out, just we've had a lot of people asking lately about using KBase for genome announcements. We're going to debut this in a public way next week, but if you see in this in the Q&A document underneath the slides I shared, there is this genome announcement blog draft. And so we're going to post this online next week, but it basically lays out in kind of a blog style narrative approach how you can use KBase to make the most out of your genome announcements, right? Not just to do the basics, but also to really further what is often contained in these announcements, and use KBase as a sharing resource to give access to your genomes and your isolates and drafts that you've made. So just would encourage everyone to take a look through this. Like I said, we're going to post it online and of course share it with all of you next week. But since y'all are the people that are coming onto our webinar, we want to give you early access to the new stuff that we're putting out there. So if you're thinking about doing a genome announcement, or would encourage you to take a look at this document. What else here? So let's start from kind of the top of getting into KBase. So to do that, we go to kbase.us, and that will take you to our home page here. This just has some general information about KBase. I'll point out a few resources on this website that you might find valuable. The first, as I mentioned, is you can check out our project paper that was published in Nature Biotechnology in 2018. It's got it's got a lot of useful information and a lot of the general overview stuff that I've covered about KBase in here. So, and of course, if you use KBase, we encourage you to cite this paper. If you ever, maybe you're thinking about using KBase for a certain project and you don't know if it's the right choice, or you want to know if there are people to connect with around, doing some analysis, we just encourage you to reach out to us using this engage@kbase.us email. Whether it's about collaboration, it's about you want to do a workshop or webinar for your specific group, and so on and so forth. This is a great way you can get in touch with us about helping. Yeah, how can we help you? So a few useful resources on our website. First of all, if you haven't signed up for an account, you can click this sign up button at the top, and that will allow you to create an account using either an existing Google account, if you have a Globus account, or an OrcID account. It allows you to create a KBase account with one of those authentication systems. But some other useful resources, I showed this briefly in the slides, but if you go into our research tab, you can see our project paper, but I encourage everyone to look at the publications that are already coming out using KBase for their research. This number is growing all the time. It's always exciting for me to see, every time I get one of those alerts that somebody has published something that cites our paper or uses our system. We're seeing people use the system for everything from medical biology to environmental and energy topics to metagenomics analysis, microbiome analysis. It really runs the gamut. So we're always excited to see how our users are using the system and pushing it in different directions. So one example I just wanted to highlight from some of our colleagues at Oak Ridge actually is, they're doing what we're encouraging folks to do. This was like a draft genome announcement that they had done from an isolated from the roots of a Populus deltoides. The article itself links to the narrative here, and so we really just again want to encourage people to do this. That if you're going to go through the trouble of using the system, make your narratives public once you've published them, and give other users the ability to access them, because what this allows users to do is make copies and analyze the data on their own with the data that you've shared. So you can see here where this user Dale Pelletier has shared their narrative. So it's a very good practice that we want to encourage you all to do. But anyway, this is just to say, check out what other users are doing and you might get some inspiration. One thing I wanted to point out is this webinar we're doing today is one of many that we have coming up. So if you go to the About section and go to Webinars here, you can see all of the upcoming webinars that we have. If you haven't signed up for these already, would encourage you to. But you can also find recordings of our previous webinars as well. So we often get questions like, where's the recording at? And so we put them all up here on this webinars page. Another helpful resource is, I'll just briefly go to our help board. So often, users need some help, right? Maybe you get an error, maybe you can't figure out why something is behaving the way it is. We encourage you to create a help board account and make tickets on there. And one of our staff will come and help you triage the ticket, help solve it, and if it's something on our end, it helps us fix these issues as they arise. So please, in addition to creating your KBase account, go ahead and create a help board account, because you may need to use it. And yeah, this just gives us the ability to, you can share your narrative with us on the help board. We can look at the error, we can look at your data and try to figure out what's going on. Let's see, anything else? Just a general kind of update about our website. We are totally redoing it right now. We're expecting that sometime this summer we'll be able to launch our brand new homepage. We're going to launch a new documentation page that has all of these guides and FAQs that we've put, but makes them a little more parsable and accessible. So we'll be debuting that soon. But yeah, this summer we're going to roll out a whole new homepage, which we're very excited about, where we'll have more of this kind of content like blog posts, videos, interviews with users, right? Maybe you're using KBase to do something that you want us to help highlight you and your work. We would love to do that and work with you on that. So just some exciting things coming down the pipeline. So let's sign into KBase and start to see what the actual system looks like. So from kbase.us, I click sign in there. I've got a Globus account and sign in here. And the first place that it takes me to is a place called the dashboard. So the dashboard is pretty much home base for getting to the rest of the system. So we'll talk a little bit about the dashboard and some of these other tabs on the side here later on. But I want to go ahead and get us into the narrative interface, because that's really where the work happens. So you can see this dashboard, it's got stuff like your narratives, tutorial narratives. Well, let's see what a narrative is first before we get into all of that. So I go to this blue button here on the side, I can click new narrative, and it starts to spin up creating a new narrative for me. And so here you can see a blank, untitled, brand new narrative. So let's walk you through some of the different components of it. At the top here, we have our toolbar. So we can do things like, go back to the dashboard. We can give it a name, like my first narrative. Spell it correctly. And it's taking a minute to save it there. So anyway, you can name it. It's got access to the help board, access to sharing, save your user account, all this kind of stuff you can get at the toolbar level. Below the toolbar, you have some panels to the left-hand side. The first is the data panel. So I want to add some data. I click this button and it opens up a data slide out with some different tabs for different ways to access data. Starting from the right-hand side, we have the import tab. This is where you can drag and drop your data in. So I'll show you more details about doing that later. We've got some example data if you just want to, try something out, some tool. Our public data here is where you can access all of the genomes from RefSeq, for instance. You can also access genomes from JGI's microcosm or phytosome there as well. And you see I've got my data, which is of course the data that I've uploaded into KBase, as well as data that's shared with me. And that will take a long time to load because there it goes. I've got quite a lot of data shared with my account. So anything that you share with other users or other users share with you will appear here. Below the data panel, we have our apps panel. So each of these categories hosts some number of applications and programs that are in KBase that you can run against your data. So I unscrolled this here, and you can see lots of different apps that I can do for comparative genomics. So let's just give you a quick example of how running apps against data works. I'll go to add data. Let's just find a genome here really quickly. Shiwanella. So I type that in. Oh, let's anyway, any of these will do really. So you can see I can add the data into my narrative. It very quickly loads into the data panel. If I click the name of it, it then loads it into this middle section here called the narrative. So this middle part, the majority of the screen here is the narrative itself. So I've got my genome in my data panel, and I want to run some app. Maybe I want to build a phylogenetic tree. So I can search for a tool that will allow me to do that. Insert genome into species tree here. And it's taking a minute here for it to load up the stuff into the actual narrative, but here we go. Let's see. Seems to be a little fussy right now. Yeah, error. That's great. So it may have been just something about the way that this narrative loaded, but let me just try to turn it off and turn it back on again. Just like, oh, here we go. I'm getting timeout connecting to KBase. So I must have had some network timeout issue here. I'm gonna refresh. Oh, that's not good. Let me see here. Hearing some reports, others having issues at home. Let's try to spin something up. Yeah, okay, getting some more information in the chat. Everyone's having problems. Awesome. That's what we like to see happen here. Let me just, yeah, a lot of people are trying to get online. All right, let's see here. Normally we don't have problems even with this many users with yeah, exactly. COVID-19 issue. That's right, Luca. With everybody is working at home at the same time, this is kind of what happens. Appreciate y'all hanging in here. We'll get it working here in just a second. Let's see if I can't though while we're all right. Let's just jump into this narrative that I had going here. So maybe we can roll through this here. All right, so hopefully we'll get the system back online shortly. But I wanted to just, the whole point of this webinar today is to take you all through what you can do as far as isolate analysis in KBase. How do you draft an isolate genome? How do you do some comparison between that genome and other genomes in the system? Basically, we want to take a concrete example of doing some analysis in KBase and teach you some information about how to use the system in that context. So you see, we've got a couple training goals with this this analysis, right? We want to show you how to import your data into KBase. We want to show you how to load public reference data. We want you to be able to do QC and QA on your sequencing reads. We want to assemble and annotate those reads into draft genomes. And we want to then look at some features of interest in those genomes for comparative analysis and do some of that comparative analysis. And then we want to place that genome into a phylogenetic context using reference data. So let me see. So it's giving me that same error here. I'm just going to keep proceeding until I get notification that the system is back online, since it's already kind of loaded in here. Yeah, so let's go down. So the kind of background of this is some research that came out of Oak Ridge from DOE a couple years back, where our researchers here were able to identify a tube gene cluster associated with mercury methylation from anaerobic microbes. So this is, of course, very important. You know, some of the sites that DOE manage has mercury contamination. So understanding how mercury is methylated by microbial systems might tell us something about how that mercury then moves up trophic levels, or you know, how does it become into a form that is can be accumulated by biological systems. So this research was done, and they identified this 2-gene cluster from some different delta proteobacteria, HGCA, HGCB. You can, you know, once the system gets back online, you guys can check out this paper and read a little bit more about it. All right. So yeah, basically what we want to do in this narrative is we want to take some, you know, brand new, you know, right off of the sequencer sets of isolate reads. We want to assemble, annotate them. We want to see basically, do these new organisms have this 2-gene cluster in them or not. So yeah, let's keep going down. So I was talking a little bit about you know, the first steps being getting that data into KBase. So there's a couple of different ways you can do that. The first is uploading and importing from the staging area. This was yeah, this section here is what we call the staging area. So after you drag and drop it, you can then import it into your narrative. This narrative has lots of little GIFs here that play on a loop to show you just kind of the point and click motion, and we use a little movie magic to make it go faster. But yeah, you can see here where, you know, it's dragging, dropping it, selecting that this is a GenBank genome, for instance, and importing it this kind of way. So that's one of the most common ways to get data into the system. I talked a little bit about the public tab earlier, where you can go and access public data and then add it to the system there. So that's important here because we, for instance, we're comparing these new draft isolates to some published isolates out there. So that's how we add those later on in this narrative, is just by going into the public data tab. You can also import reads directly from the NCBI SRA. So if I go down here, there's a specific tool that we have here called import SRA files as reads from web. You can access that via the upload panel and the apps panel here. If you go down, you can see import SRA file as reads from web. It's a little tricky to use this tool, so we've tried to annotate some of the images here about where exactly should you be looking at on the NCBI website. So you do have to make sure you're going to the run browser in the SRA, and that the link you're going to drop comes from this data access panel here, where you can then grab the link to the FTP. And importantly, you do need to know the insert read length average and the standard deviation to import these. So oh yeah, it's probably not going to connect to the kernel right now. So anyway, you can use these links. You can use this information later on to import some reads from SRA into the system. Finally, we have the ability to add data directly from JGI's Genome Portal, at least isolate reads. You can, we'll talk about the search panel later, but you've got the ability to search directly against the Genome Portal and import those reads into your narrative for analysis. So yeah, you can see this GIF running there to show you how to do that. Okay, so what we did is we did import some reads from SRA. So we found this delta proteobacteria, Desulfoluna, butyrate oxidins in SL71. This was, I think when I started to draft this this narrative, this was just the latest delta proteobacteria that was on SRA. So I found it, imported it into my narrative here. So these are some reads, untrimmed, no quality control, that we want to assemble into a genome. So the next section here, we've got some quality assessment, trimming, assembly, and annotation that we're going to do. So the first thing we want to do is do some quality assessment of our reads. So we have a very popular tool in the system to do that, FastQC, which is a very, very commonly used tool because it makes a nice printable output report here that we can see. So what we see in this this sample is fairly typical for Illumina reads, just the general pattern of the sequence quality sort of falling off as we get closer to the end of the read. This is very common to see this with Illumina data, and so it indicates some need for us to do some quality trimming towards the end of the read position. We can see information about the quality per tile. So this tells us something about if there's any bubbles or errors with particular cells in the sequencer. And yeah, overall, this just gives you some basis of comparison of your reads to a theoretical standard. So sometimes you have to be careful in interpreting the results of FastQC. Sometimes, for instance, I'm seeing these observed this Kamer content. This could be just because of adapters that were unable to be removed. It's hard to say exactly, but overall, this gives you some information that you can make decisions about how you need to triage your reads before you assemble them, because with assembly, it's I guess a garbage in, garbage out kind of thing. If you put in low quality reads and assemble them, they may complete, but your ability to then infer what's inside of the genome is affected by using those poor quality reads. I think I linked to it. Yeah, there's a great tutorial and FAQ for FastQC. I think this is on the University of Michigan website, or Michigan State. So they've got a lot of information already there about interpreting the statistics on FastQC. So we just linked to that at the top of this. Yeah, I'm sorry that the website is crashing for folks. I'm waiting for the signal that it's back online and everything is working here. So let's keep going. So one of the things, like I said, is we saw the need to do some trimming to bring the quality of reads into a more high quality Fred scores, to trim off any low quality Fred score reads in there. So let's see if I can, yeah, it's not gonna let me show you the configuration, but essentially the default values of Trim-O-Matic will trim any reads that have a lower than quality score 15, which is pretty low quality as far as Fred scores, go up to 36. So yeah, that's it's going to trim off those low quality reads, and well, more or less what we see is overall the quality score improving. Yeah, so yeah, we see, I guess I'll go to the other one to do some comparison. So here is our pre-trimmed reads, and here is after trimming. There's still a little bit of low quality on the front. This may just because of some issue with the sequencer, hard to say, and it looks like there's a difference even between the forward and reverse strand, where the reverse had much higher quality in the earlier positions of the reads. So that's the effect of doing some FastQC. Yeah, so Matt, seeing your question in the group chat, yes, so yes, you can upload the FastA files into KBase to do FastQC against them. So now we have some reads that we think are in a better position to do assembly on. So we're going to move on to Denovo Assembly of reads. So in this example, we don't do much comparative assembly for what it's worth. And in fact, this is like maybe this is just a general trend in assembling prokaryotic isolate reads, is that a lot of the open source assemblers out there have been pretty much dominated now by one assembler called Spades, which has just been consistently updated and upgraded over the years. So that we have a lot of assemblers on the system, but just generally speaking, if you're working with draft isolates, just in our experience, Spades is almost certainly going to work the best. We do have, it might be challenging to access it right now. Oh, maybe it's working again here. Maybe the system is working again. I'm gonna take the risk and refresh this narrative shortly. But so if you want some information on like doing a little more in-depth comparative assembly of prokaryotes, in the dashboard, if you go under tutorial narratives, you'll first of all see all of our tutorials. But this first one here, assemble and annotate prokaryotic genome, goes into greater detail about some of the strategic decisions you have to make about genome assembly. Okay, it's looking like the system is working again. So I am going to refresh this narrative and hope for the best here. Maybe this one has already loaded. No, okay. Close these down here. All right, yeah, we're getting indication that it's working. It's going to take a little second since I'm reconnecting to the kernel to get back into this narrative, but this is a good sign. Yeah, so here's just this example narrative. There's actually a publication associated with this as well, a protocol paper that we wrote, and this just kind of goes into some of the details about comparative assembly. So if you want to do some comparison against other assemblers, you can do that. More than likely, you might just be trying to compare assemblies from the same assembler. So I just reloaded it right when it loaded there. Yeah, so you may, right, if you're configuring spades, let me go down to it here. Lost my place. Here we go. So if you're configuring spades, by default, spades does a lot of automated configuration. So it sets k-mer values automatically. For instance, you can add custom k-mer sizes. You can set at what the minimum config length is. So you can filter out any configs that are at a length below whatever you set here. So yeah, you can configure these assemblers, do some comparative assembly between them to see which one generates the better statistics. And so when the assembly is done, you get a nice report here telling you about the statistics of that assembly. You get your N50 values, L50 values, and number of predicted genes and so on, with lots of other statistics you can get about the assembly. So once you've assembled your reads, what will come out of that is a assembly object. So here you can see, it's created this object here, desulfoluna butyroxidins spades here. So I can click that and it will load that assembly, and you can just see some information about the assembly in terms of lengths, GC content, etc. So with all of the data types in KBase, you can do this kind of second level analysis. Each data type has its own viewer with statistics. If I go up to the reads here, this is the data viewer for a reads type object. You can even see some information here saying this is a paired end library, statistics, etc. So after we have an assembly, right, I can then do some annotation with one of our annotation tools. So we have two different annotation tools in the system right now for prokaryotic isolates. One is based off of the RAST toolkit, and the other is based off of PROCA, which is a prokaryote-specific annotation tool. So you can annotate your assembly with either one of these annotation tools, and so that will load up the some statistics about each one of these annotation jobs. You can see that after I run the annotation here, I can select the assembly, set some parameters about it, and after I run and I get the annotation completed, I get a nice little report here telling me about how many coding genes it found, how many non-coding genes, hypothetical functions, and so on that I get with this annotation. You get similar kind of report with PRACA. You'll just notice that even though we're effectively doing the same thing, and it generates the same type of object, this genome here. We get different reports based on different apps, and that just has to do with the fact that all of these apps are third-party tools that we're bringing into the system. So you may see differences between reports. We're trying to do some standardization of those things just to make it easier and more streamlined and uniform. But just, if you're wondering why that's the case, it's just that these different tools bring different reports and such with them. So yeah, this is what we can use to annotate our genomes. I've got some notes in here about some differences between how these different annotation apps go about identifying and annotating different genes. So yeah, if you can read that later on, but we'll keep moving forward here. So yeah, what we've done is some quality control, we've done assembly, we've done annotation, and so now we have genomes, multiple genomes, one from RAST, one from PRACA, that we can use for downstream analysis. So here I'm looking at the genome object viewer here, so I get some overview information. If I want to look at the features of this genome, I can go to Browse Features, and I can see different functions. I can do some filtering here, like something in there, and yeah, it will start to filter this list of features. If I then, let's say I want to go into this gene, see something more about it, I can click the name of that gene, and you'll see it comes up with some viewer about the position in the genome. I get information about the protein translation sequence and the DNA sequence. So yeah, I can start to actually look into what does this genome have inside of it, and how do I compare that to other organisms. Seeing some stuff into the chat here. Okay, yeah. Thanks for sharing that, Zach. Yeah, you can access this narrative, of course, from the link in the Q&A doc here, or via that link that Zach just dropped into the chat. All right, so yeah, we've got genomes here. Let's see how we can go and then do some more analysis. Yeah, one last tool here. A very popular tool, CheckM. You can use CheckM to do some quality assessment of genomes. So we can see that this is a pretty high quality genome here. I can see that it's 100% complete, has less or just about 1% contamination. So this is more often used if you're trying to look at several genomes, right? Maybe you have several mags from a community sample that you want to do assessment on, but we can also do it for isolates as well here. Another new tool that we have is a genome visualization tool built out of another open source platform called CGView. Oops, just close it there. This is right now, this is in a static form. It's just a way to get a kind of overview image of your whole circular genome here. You can do some configuration to get different annotations on the map. But this is just a new tool that we've just put on the system so that, if you're doing your genome announcement or something like that, you can also get some graphical representation. One thing you'll see here is, like, in the results section in the report, below the report, there are just some different links. So if I wanted to download, scaled vector file for this, or just a JPEG or PNG, the app produces all of these files that, if I click one of these, it'll start to download it on my system. You can download these files. You can also download data from the data panel, just anything you hover over. If you see these three dots, you can then see some more options here, and one of those is to export and download data. That one is not a great example because it's internal KBase file type, but yeah, I'll maybe take this example with these reads. You can see that I can download those as FASTQ file directly off the system. So there's multiple ways that KBase is feeding you up your results for you to take and then put into your paper, or maybe you have some other tool that we don't have and you want to analyze them off system. You can do that as well. So that was one genome that we pulled in the system. I pulled in another set of reads from JGI and added them to this narrative, and I did the same thing there as well, do some assembly. These were already filtered and quality controlled, so I just fed them directly into the assembly pipeline and did some annotation. And so this was just two input genomes that we wanted to use for some comparison, some new draft isolates that we wanted to use as comparison against some known isolates out there. So, okay, we have gotten these new draft genomes added. We assembled them, annotated them. We want to start to do some serious analysis to see what genetic content is inside of these genomes. Specifically, we want to see, do they have this 2-gene cluster of HGCA and HGCB? That would indicate that they have the ability to methylate mercury. So what we did is, if I go back to the top, actually, maybe I'll just really quickly show this. Go all the way to the top here. So yeah, this is from this paper in Science. You can see that these were some known isolates that that the initial group was able to demonstrate had this 2-gene cluster. So we want to take these as the basis of investigating against our new draft isolates. So we want to add a couple of these into the system and try to use them as comparisons against our new data. So what we're going to do is we add them into this narrative, and then we want to isolate these features so that we can compare against our new genomes that we just assembled and annotated on the system. So scroll all the way down. And you can see here I've loaded 4 of these public genomes that were referenced in that diagram above. And now I want to build do what's called building a feature set from them. So all of these genomes have this HGCA and HGCB gene. I want to build a feature set from those so that I can search for them against my new isolates. So what do? I load this build feature set from genome. So these are in the utilities category. So you can see build feature set from genome here. What I do is I've loaded this genome of interest, and I searched basically the annotation or the name of this gene in this genome, and I selected those genes, and I said, okay, I want to find this HGCA. This is what it's tagged as. I can select it, and then I can build this output feature set. So what I did here is I built feature sets from all of these different genomes. and I merged them into a single feature set. So these are all of the HGCA genes that I isolated from each of these genomes. I have put them into this object called a feature set that says combined HGCA. And I did this for the HGCB gene as well. So similarly built genome set, built feature set from these 4 genomes and merged them into this combined HGCB. So here's another graphic from that original paper where they did a multiple sequence alignments, and they identify these, particular regions of interest that are highly conserved, across all of these different species. So we want to how do we do that in KBase? So we are able to construct a multiple sequence alignment from these feature sets. So I input this combined HGCA feature set, I give it some description, and I'm able to construct a multiple sequence alignment that I can then use later down the analysis. So I get this clustal file here, showing the alignment between all of these genes. You can see this, highly conserved sequence that they identified in these other example species. So next, I want to do some searching. So I've got my multiple sequence alignment from all of these known draft isolates, and I want to search against this new genome I've got to see if it has it. And sure enough, when I do that search, I do get a hit here. And similarly, when I search against for the HGCB, I also get a hit here as well. So that to me says something good. That means okay, these new drafts, they do have the 2 gene cluster that we would associate with methylating mercury, and we can see, actually they are indeed right next to each other in the genome because of the number of their gene ID there, HGCB A and B corresponding to CDS 26 and 27 respectively. Similarly, this is just showing you there's some other tools you can do this sort of protein searching with. So yeah, the tools that I just used were Muscle and then Hammer. So if you go to the sequence analysis section here, you'll see these tools available. So there's a couple of different ones in there that you can use. One interesting thing to note, and I think this happens later on. Yeah, I had some errors. This is something we need to fix. But if there are no hits found, this will generate an error. So that's just something to take note of. If you're doing this and you're just getting errors all the time, it may not be a issue of the app. It's a failure to report that it has no hits, and instead it's generating this error. So that's just one known thing that we need to fix. Another really useful tool is Paper Blast. So maybe you have a gene of interest, especially maybe if it's a hypothetical protein, and you don't know what it is and you want some kind of reference, you can actually search against the reference literature right inside of KBase. So here I just took that protein translation sequence and pasted it into this tool and hit run. And sure enough, I get some interesting set of papers, referring to this this 2 gene cluster. So yeah, this is just kind of an interesting way to search against the published literature to see if there are some clues, maybe if you're looking for what does this gene do. All right, just checking the chat here. Yes, paper blast, very cool. So that's what we did in this section here. I know it went kind of fast. Is that we isolated some features from some known draft genomes. We searched for those features in our new draft genomes, and we found some hits. That's more or less what we just accomplished, is that we were able to locate these genes within our new genome. So now what we want to do is to kind of, so that's right, we zoomed in as to the gene level. Now we're going to kind of zoom out to looking at the whole feature profile and domain annotation of these genomes, to kind of look at the features as a whole. So what I did here is first of all, I built a genome set. So what I did is I took all of those reference genomes, and I took my two new genomes and I put them together in one single genome set here. What this allows us to do is, like, some of these tools in KBase, they operate on individual data objects, or they operate on set objects. So if we can build a set from all these genomes and run some kind of batch processes against them. So what we do here is a very long running process. This probably takes longer than any tool in KBase because it's pulls from huge reference databases, is this annotate domains in a genome set. So what we did here is we're taking this set of genomes and we want to annotate them with some widely used domain libraries. So just to be more concrete, let me, I'll open up the individual version of this app, and you can see in some greater detail what those domain libraries are. So this is, we have two different apps here. One is annotate domains in a genome set. One is annotate domains in a genome. You see we've got these different domain libraries here. By default, it will run all domain libraries, and so that's what actually this tool does, is it just annotates with all domain libraries. But if you want to be more specific, you want to use pfam, tigerfam, conserved orthologous groups, whatever, you can select those domains. And all this does is adds another layer of information on top of the annotated functions on your genome that relates them to protein domain families and libraries of those families, so that you can understand maybe some more about the what your genome can and can't do. So this is a necessary precursor step to what we do next, which is view function profile for genomes. So we annotated all these genomes, and we then are able to compare the presence or absence of genes organized into these different domain families. So I can blow this up into a much larger report, and you can see here, this is like quite a lot of information. So this is really kind of doing an all versus all type perspective, but you can do some filtering to make this a little more concrete. When you open the, if I go to comparative genomics here and go to the bottom, I'll see this view function profile for genomes. When I'm setting up this app, you can select which specific domains namespace you want it to generate the feature profile for. You can actually just set specific domains. So for instance, if I, I've got COG selected here, I can select only to show, some sub-selection of these domains that I'm interested in. And so this is what you would do in practice, if you were looking for some specific feature. You would filter, based on what what its general function is. So you can build more specific function profiles in that way. But all we did in this one is just to show you kind of like all versus all, what that looks like in the system. So this is what you would do if you wanted to, do some kind of comparison between multiple genomes. And we'll go into, with some of the later webinars, we'll go into greater detail about that. But this is just to show you an example now. So finally, maybe we have some draft isolate and we want to understand what it's related to. We want to put it into some phylogenetic context to see how it might relate to what we already know out there. So we've got a couple different tools that you can use to accomplish that. I started to show this earlier when things were messing up, but we have this insert set of genomes into species tree tool. This similarly, it operates on a set, so I can drop the set in there. There's a version that also uses individual genomes. So if you wanted to put an individual genome into a species tree, you can do that as well. But this generates a tree using the fast tree algorithm, and it uses a subselection of conserved orthologous groups to measure some dissimilarity between each of these genomes. So there's a subselection of like 49 protein domains that it uses for making these organization of the tree. But yeah, this just shows you that you can generate a tree pretty rapidly on system, and you can download all this tree as a NUIC file. So if you want to do some annotation or some organization filtering outside of KBase, you can do that. Or you can just grab the PNG copy of the tree and put it in your paper. The other tool that we have, which is a new one that we've added on the system, is this GTDB. So this uses the newly released genome taxonomy database that's out there. And it similarly, we're able to add this whole set of genomes. And this doesn't generate a tree for us, but it does do some useful computation and gives us some statistics of value. So I'm going to click View Report in separate window here, where I can just see a lot of information about the input genome. I can see where it placed it taxonomically. That's with these new ones, perhaps unsurprisingly, it doesn't get down to the species level because these are brand new, isolates that I don't even think have been put into NCBI yet. But so we get some classification. We get some information about ANI. So, with these known genomes, unsurprisingly, we're getting 100% average nucleotide identity scores because they match hits that already exist in the reference database. So yeah, GTDBK is a great tool for doing phylogeny and taxonomy. It's very, very valuable, especially if you are looking at metagenomic data or mags, you can get some placement from your bins using that. But in this context, it's just to, you know, if we had a isolate that we didn't know where it was taxonomically speaking, we could do some initial placement for it. And let's see, where did, I think this is the last thing I did in this narrative. Yeah. So just to give a recap, what did we do here? I'll fly all the way back up to the top here. If I can grab my scroll here. So we looked at some of the different ways you can load your data into KBase and how you can access public reference data. We showed some of the quality analysis and control tools for the system. We showed you how to construct isolate draft isolate genomes by assembling and annotating them, and we showed you how to dive into those genomes to look at some different features, compare them to other genomes on the system. And we showed you some tools that you can use to place those genomes into a phylogenetic context. So this tutorial is something that we're actively updating and growing as we add new tools. So you can see I'm kind of trying to keep tabs of the new stuff that I add. You all have access to this now. You can access it from this Q&A document, but we'll put it online for public access shortly. Thanks again to everyone for coming on. We got another 10 minutes here where I can go into some more detail or help answer some more questions. And of course, we will share this recording with everybody after the webinar is done today. So so you can look forward to that in your email. Let's see, what else can I show? Oh, I didn't go through kind of like the full intro aspect. I started to run into those issues and jumped right into the narrative. Yeah, so you can do chloroplast or mitochondrial assembly on system. You can do those as well. So yeah, thanks again, everybody. Really appreciate and thanks for being patient as we worked through some of our technical difficulties. I have an app related to structural genomics which has autodoc support. Should you join a different session for that? They might cover that in some of the. Oh yeah, I should show you all the upcoming webinars to do some advertising of that if you haven't signed up for those. So, and we'll probably cover, we don't have, we're actually putting in some tools for doing structural analysis of proteins, doing auto docking. We don't necessarily have a lot of that stuff available right now. It's up in beta. But if you come on to this functional genomics or this microbiome metabolic modeling, they'll talk a little bit about some of those tools or more so where they're going to be integrating them. So yeah, you'll be able to find this video up on this kbase.us/webinars page. We put them all under this previous webinars section, so you can for sure come and sign up or view past webinars. Oh yeah, I just wanted to show you all just some of the stuff about the dashboard since I had to jump away from that. So everything that we did, you know, anything you do in KBase, you'll see it stored in this Your Narrative section up at the top, so you can see all of the narratives that I've worked on up here. You can see some of our tutorial narratives in the second section. Narratives shared with you, public narratives, and so on. To to get to the organizations that I referenced earlier, you can go to this orgs page where you can view existing organizations or create your own. If you want to look at all of the apps that KBase has available, you can go into the app catalog here. And this allows you to go through the different categories and check it out and see the different tools that we have on the system. With any of these tools, you can look at more information about them just by clicking on them, and it'll tell you more about what each of these apps does and what the parameters mean. So, for instance, we just showed this view function profile, and we can get a lot of extra information about what this app does and what all the parameters mean. Talked about searching for reference data earlier. So we have the search feature here. If I type in something to search, hit enter, it will look for stuff here, and it will start to identify both narratives that may contain the data I'm interested in, also reference data that matches it. I can change the resolution of the list, and maybe I want to select multiple of these and move them into a narrative. I can do that. Move them into a new narrative, copy into an existing narrative, so forth and so on. I didn't show it while I was reviewing the previous narrative, but you can also switch the tab to JGI here, where you can search against the JGI Genome Portal, and here you can see all of the hits for Geobacter. And similarly, you can move this data into your staging area, then into your narratives. The jobs section here, this tells me about the different jobs that I've run. And so, if you have tons of different narratives open, I've run hundreds of jobs, and you're trying to manage all of those, this is the way to do it rather than in the narrative itself. So you can see the status. If there's errors, I can if I hit more information, I can get information about the job log here, so I can just see every line of execution for a job. So that's very useful. With the account section here, you can help manage your account. We always just encourage folks to try to add as much information as you can. You can also, people often have multiple accounts because maybe they've created one and then they forgot their username or something. You can actually link accounts here. So if you have multiple accounts, you can link them to your KBase account. And what that will do is share access to your data and narratives across all of your accounts. Finally, we've got the feeds section here. This is still a work in progress, but the function of this is to tell you about, to give you some notifications about what's happening in your narratives or inside of your groups. So you can see I'm getting updates about people trying to join different groups and so forth and so on. So yeah, these are some of just the different resources that you can access from the dashboard page. So hopefully that gives you some insight into all of the things that you can do. So we'll quickly go back to the Q&A document here. I'll send, in addition to the recording for this webinar, I will send this feedback form here. If you all wanted to take a minute and give us some feedback about the webinar, that's very helpful always. And of course, anything that we showed you today is on this this document. If you are on social media, just really want to encourage everyone to follow us at DOE KBase on Twitter. We announce these kind of webinars and training events. We announce features, we share new publications, all that kind of stuff. on our Twitter, and increasingly we've had more and more people telling us that they like KBase on social media. So we always appreciate that. So if you ever want to tag us or say hello or ask a question, you can do that on social media as well. So please follow us there. And let's see if there's any unopened, unresolved questions here. Seeing quite a lot of stuff. Let's see if I can. Oh man, yeah, quite a lot here. I think somebody is adding G blocks. Actually, maybe I can go to the catalog. It might be in beta. So all of the apps that are shown here, or if you're in the actual narrative interface, these are all showing you the released apps by default. But you can see here I can toggle between release and beta versions. So if you want to help us beta test apps, you can always see what we're working on here. So yeah, you can see we do have G blocks, so you can trim those MSA. Actually, I think this is released, if I'm not, yeah, so there is a released version. So if you want to trim those multiple sequence alignments down to only the shared sequence, you can do that. Let's see, what other questions we have here. Does it have to be the full protein sequence? Can it be the putative protein name or only the partial sequence? You can only right now, so I would only recommend doing the full search. You can't do just the name. And as far as like minimum sequence identity goes, it's one of these things where you kind of have to use some best judgment. If you're looking at it really depends, because on what the conserved sequence structure is going to look like. But I would say that you should be looking for ones with fairly high matches and identity percentages. If you're getting like 20-30%, it's probably not the same, but you never know, again, because it depends on the length of the sequence. It depends on how much variation there is between those sequences. You could, of course, if you had a highly conserved section or something like that, you could, of course, you could plug those into the paper blast, but your results may vary depending on what it is you're searching and so on. Can domain search be automatically linked to the annotation? So I think this kind of gets into some of the questions around knowledge engine, which we didn't show you anything about that today. We'll once we have some of those tools released into alpha, we'll do some webinars about them. But I mean, essentially, yes, what you're talking about is what we want to create the tools for. So right now, all of these things have to be kind of sequenced together and placed into a workflow to make them sensible. The inputs and outputs align and to get these findings in that way. But we are trying to develop tools that basically do what you say, where the annotations that you add to a genome are then connected to domain annotation databases, and the system maybe informs you like when those annotation databases updated, and then a potential new annotation appears on your genome, right? Or a hypothetical feature or protein is given and assigned a new function. We at this point, our system doesn't have any of that user facing. It's what we spend a lot of time working on, but so your head is in the right place, and so we just ask that you stick with us as we work to implement some of these features onto the system for like automated and machine learning approaches, because that really is one of the underlying principles of this project, is that we are trying to develop that technology because it doesn't exist. So No, right now we can't. Yeah, so right now the domain databases are all fixed and are based off of these organized consortium like PFAM, and I'd say that like the PFAM is probably the most frequently updated of these. So that's maybe just a recommendation if you're trying to do domain annotation, is to use the PFAM library. I don't think TigerFAM is maintained anymore. Cogs, I think, was last updated in 2014. So yeah, these things slowly change, if they change at all. But we are trying to think again about how to allow users to do some curation on their data in a way that doesn't change how that data sits in our system in terms of so if you like change a annotation for a genome, it's one thing if you're doing that on your own genome, but if you're do it on a piece of data that's referencing reference data on the system, that might change things downstream. So we're trying to work through some of those questions too, because obviously you all are almost certainly trying to identify unknown functions and things like that, and so you're making your own annotations. So yes, you can download your IMG annotated genomes and upload them into KBase. But yeah, it's not redundant necessarily. JGI tends to curate its own annotations in-house, whereas some of these other tools rely on reference databases. So it wouldn't necessarily be redundant. You might find useful information by comparing the two, right? But as far as the annotations in KBase right now go, they are automated based off of the parameters that you set for RAST and PROCA. So they're not manually curated. Just trying to. As a question as new users, if there's a resource for various tools like Quasterm, etc., and descriptions given. Yeah, so this is where I recommend just diving further into the documentation. So like, for instance, Trimmomatic, I kind of glossed over it earlier as I was just trying to show something on the screen while the system was down. But you see here, I went to Trimmomatic here and I clicked these little 3 dots. I see a little information. I can click more. And that will take me into the app page that tells me a lot about the app itself, that tells me, just links me out to different resources like the manual and so forth and so on. So like, kind of our approach to a documentation is not so much like, we don't necessarily try to reinvent all the documentation for these tools. We try to make it relevant to the KBase user experience. So, for instance, with some of these tools, we hide some of the parameters just because we've made a decision to hide some of the parameters to increase likelihood of success or user experience, make that better. But if you want to understand, how to set those parameters or what they mean is in KBase, as far as KBase goes, you can go into these app pages and find out more information about all of the parameters, the inputs, and so forth and so on. You can also get access to the publications associated with them because, right, when you're doing your analysis, you don't just want to cite KBase, you also want to cite the use of these third-party tools. So yeah, I just encourage going into the app pages and looking at the documentation there, and using our tutorials. In the tutorials, we kind of try to talk a little bit about the strategy and our approach to using these tools, more so than just the point and click stuff. So if you wanted to use this for your classroom, or use it for some kind of training for your own classes, we're happy to help you with that. And I think we are actually doing some coordination right now with different groups that are trying to use KBase in the classroom. So if that describes you, please reach out to us at that engage@kbase.us email address. Yeah, lots of good questions here. Yeah, no, you could use the HitMer tool to assemble fungal genomes, but that's also, we're currently working out some bugs with it. We do have access to all of JGI's microcosm genomes. So these are all of the fungal genomes that JGI has assembled and annotated and curated. So yeah, there's 173 of these fungal genomes available. So you can do things like metabolic modeling and so on with these as well. Trying to see some multiple chats here. Lots of thank you. Yeah, thank you all so much. I really appreciate it. Yeah, the command line on the bottom right. Yes, we didn't talk about this at all, but I briefly mentioned at the top that that KBase is built on the Jupyter notebooks, so you are actually able to do coding. In addition to all these kind of point and click and GUI based tools, you are able to do scripting with Python against this data, against the tools in the narrative. So we don't have many examples of that, except for some published examples that people have used in their publications or narratives associated with their publication. We're going to try to make some of those tutorials on how to use that to do some scripting in a constructive way. But just as maybe as a kind of clue here, within each of these apps, actually, if you we call this an app cell. If you go to this 3 dots here, oops, at the top, and hit show code, you can actually see the code that this tool pulls to run, run basically what you program it to do in the graphical user interface. So it is possible to create loops and stuff like that, iterating through different parameters if you are an experienced Python person. So yeah, right now it's only Python, so you can't use R or JavaScript. It's Python 3.5. So yeah, just something to be aware of. But yes, you can code alongside doing the kind of point and click type analysis. Yeah, good question. Why might you move a genome into the staging area versus the narrative? So yeah, if I didn't make this clear earlier, yeah, sorry. I think the technical glitches prevented me from showing this. Let me maybe go and just show this really quickly in a new narrative, like what the process of upload looks like, since we didn't really we weren't able to cover that earlier with the glitch. Oh, just do new narrative. Yeah, because so the process works by you upload your data into the staging area and then into your narrative. So I was actually doing this right beforehand. Let me open up. So I've got some example reads here that I'm showing. And this is a blank narrative. I've deleted all the data from my import here. I want to import it into the interface, the staging area first. So I just drag it, drop it. Oh, it's going to go really slow, isn't it? So I drag it and drop it into the staging area first. And once this is done, well, I think I can do this really quickly just to. So you wouldn't want to do that normally, but since that was slow. But once it has completed that process, it will then show up in the bottom here. And this is where I would then select, what is it? I click this up button here, and that opens up the cell to import this into my narrative. So, this is some reads. I know it's an interleaved file because I only have one file that I'm linking to. I'd hit this run button, and this starts up the job to actually import it into the narrative. So why is it like that? Well, sometimes you might have multiple narratives, right? You may want to drag and drop all of your data at once, and then migrate those files into different narratives. That's one use case. The other, is just to kind of de-link it directly from the narrative. So, right, if you like delete your narrative, or maybe someone deletes your file from your narrative, that's a shared one or something like that, you'll still have the ability to access it according to your own Globus. One thing, this should be pretty easy for me to show here really quickly. If you have, I mentioned it earlier, but as far as bulk sharing goes on KBase, or like if you want to upload a lot of files, you may see this warning in the top here, for large files over 20 gigabytes. 20 gigabytes is sort of like the limit of Chrome and Firefox and all the different browsers for like the size of a file that you can drag and drop into here. So, of course, there are files for omics data that are much larger than that. But what you can do is connect to, sorry, I don't have it running on my laptop right now, but if I had my Globus Connect personal endpoint running on my laptop here, I could set to a file folder path on my machine or on an FTP site, whatever. And then I connect to this KBase bulk share, and then to my username, / your username. And that is your KBase endpoint. And this is the same as your staging area. So this is the file that I just added, dragged and dropped. But if I had, dozens of terabytes or something here, I could still link that to my KBase bulk share. And that also is why we have the staging area, is because you can then push your files from the staging area into the individual narratives. So hopefully that makes a little more sense about the relationship of the staging area to the narrative import, and why we don't just sort of directly, do drag and drop or something into each narrative, because you can add way more data using Globus here. So hopefully that gives some clarity. Let's see, what other questions? Looking like we've got a lot of them here. Okay, so thanks again to everybody who logged on to the webinar today. We really appreciate it. Thanks for being patient as well, as we had a few glitches there. Again, hope to see you on the system. You'll be hearing more from us as we put on new features, add new data, and all of that stuff. So please stay in touch with us on our help board, or reach out to us on Twitter, or via that engage at kbase.us email address, and let us know how we can help you get on the system. If you're running into errors, if you have questions, and so forth and so on, we're happy to help you. And we'll send around the recording to everyone shortly. All right. Thanks again. Take care.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.